The spiker box is a low-cost, effective bioamplifier that can be used to measure the neural activity of certain invertebrates such as cockroaches and crickets. The spiker box shows neural activity of organisms by displaying spikes or action potentials when plugged into an iPad or laptop. The nervous system is a network of nerve cells and fibers that transmits nerve impulses between parts of the body. The nervous system is composed of billions of neurons that help transmit electrical impulses or action potentials across the nervous system. Action potentials occur when a neuron reaches threshold. Threshold is when a big enough electrical impulse causes sodium channels along the axon of a neuron to open up. Since there are more sodium ions on the outside of a neuron, and the inside of the neuron is negative in relation to the outside, sodium ions rush into the neuron. Because sodium has a positive charge, the neuron becomes more positive and becomes depolarized. Eventually, the potassium-gated channels will open as the sodium-gated channels close. As potassium rushes into the neuron, that depolarization of the neuron is reversed and returns the neuron back to normal condition. The particular reason we used the spiker box was that it was a STEM project, which stands for Science, Technology, Engineering, and Mathematics. The science aspect of our project was learning about the nervous system of organisms and what action potentials look like. Technology was displayed through the use of the spiker box, a cutting-edge piece of technology. Engineering encompassed the first portion of our project, which was building the spiker box. When talking about the STEM project, it is important to go more in-depth on engineering because in the process of completing the spiker box, we covered a few important aspects of the engineering design cycle. For example, we didn't specify, design, or model this useful device, but we did assemble it and test it, which is still important. Moving back to the STEM project, mathematics was then shown in this experiment through the data that was collected. Because this is a brief overview on how to make your own spiker box, you should visit backyardbrains.com for a more comprehensive list of instructions and videos. To begin with, take your PCB board and solder all the pieces into the correct locations. After that, assemble the speaker and electrode. Once completed, finish building your spiker box by screwing on the plastic housing and installing the speaker and electrode. Before we could start our main experiment, our group performed an entry-level experiment to prepare us and help us achieve further understanding on how to use the spiker box efficiently and effectively. The first step in the entry-level experiment was to take a cockroach. In our case, we used golden discoid cockroaches. And then you have to place them on a slab of ice. The reason you must put the cockroach on ice is because the ice acts as an anesthetic. This causes the cockroach to stop moving around frantically. Once the cockroach has stopped moving, you must cut one of the legs off closest to the body of the cockroach. But don't worry, because the cockroach will still live. Also, make sure that when you cut the leg off, that you cut it as close to the body as possible. This is very important because if you don't do this, then the leg won't be able to be used for experimentation. After cutting off the leg, place the cockroach leg onto the spiker box. Once on the spiker box, place one of the electrodes on the coxa and the other electrode on the femur. You should then hook up your spiker box with the correct cord to an iPad or computer. In order to collect data, we suggest that you plug the spiker box into a computer. Although there are other devices you can use to record and analyze data, such as an iPad or an Android, computers are the preferable choice because it is much more user-friendly and easier to analyze. Here's a clip of what data looks like on a computer. You can put data above and below each other to compare and contrast what data you collected. In order to analyze data on your computer, it is recommended to download and install the software called Audacity. But before you can see spikes, you are going to need to modify the default settings. If you are interested, the settings can be found at backyardbrains.com. Now that your cockroach leg is in place and your computer is plugged in, you can now observe spikes. As part of our entry-level experiment, we blew lightly and eventually vigorously on the cockroach leg. This in return resulted in an increased amount of neural activity, which means more spikes. After observing what happened to the neural activity of the leg after blowing on it, we then proceeded to test the new stimulant. The new stimulant was gently touching the barbs on the leg of the cockroach. This, just like blowing, resulted in an increased amount of spikes. This concluded our entry-level experiment. Now we will proceed to the advanced level experiment that our group has decided to undertake. To complete this experiment, we needed 2 spiker boxes, 2 cockroach legs, and petroleum jelly or Vaseline. The purpose of this experiment was to find out how long it would take for a cockroach leg deprived of oxygen to stop showing spikes and to see how long it took for the spikes to return after allowing oxygen back in. The first step in this experiment was the same as the entry-level experiment, and it was to prep the cockroach leg for the spiker box. After the prepping of the legs is complete, place each leg on the spiker box. Next, coat one leg with petroleum jelly but leave the other one untouched. It is preferable to have 2 spiker boxes and 2 computers because, as you can see in our lab setup, we have the control leg on the right side and the experimentation leg on the left side. This allows us to compare data in real time. Due to the inconsistent data that was collected during experimentation, we can understand through knowledge that the petroleum jelly should have affected the spikes because without oxygen, the neurons can't function properly and the sodium-potassium pump won't be present. Thus, the action potential couldn't be created. Without the action potential, there won't be any spikes. Our leg stops spiking within 10 minutes, but don't be alarmed if it takes longer, because every cockroach leg is different. As you can see in this picture, the experimentation leg, covered in Vaseline, stopped producing spikes, while our control group was still producing spikes. Through the extensive building and experimentation process, we have learned a lot about how the nervous system works and became increasingly skillful with the spiker box.